in today's online open lecture. My name is Guangxue Wu from University of Galway, Ireland. I'm the coordinator for this Marine Career doctoral network, project. So this project is about leveraging anaerobic digestion through that environmental stresses. So this is the logo for our project. And also, if you are interested in other information, you can find, from this website. So anyway, for this series lectures, here is the detailed that information. We will have the following 6 lectures. I think also this information can be found online. So anyway, if you know someone else may be interested in the topics, you can send an email to Yuyin Wang. She's the project officer, manager of our project. And also this project, we have that 8 beneficiaries from different countries. So maybe first I just introduce about some brief introduce about the project information. For this project, we designed this logo, the lead, this just the AD and that leveraging, and for this different curves like a book or view that shows for anaerobic performance, maybe this normal condition and also under stressed conditions, and we hope that we can apply different technologies to recover this performance, and also that different colors for this one, this corresponding to that different sustainable development goals. So this is the concept for this logo. And also the following several slides shows the project information, especially for those supported by this project. I think if you are interested, maybe later you can just check by yourself. I will not introduce this in details. So this is the project, this is the research concept, and also one aspect is that PhD training, and also this the different tasks. We have 14 that projects for 14 that PhD students. Also this the project management. So every that beneficiaries is participating in different aspect for this management. I will not talk this in details. So now I just goes to today's talk. Today I will talk about 4 aspects. One is mainly about this principle, about this AD, and also its metabolic pathways, and also the following 2 sections. I just try to talk our research outcomes by using that RK selection theory, how to apply this principle to that AD. And finally, I will talk some concepts for these concepts that may be summarized in other published papers, and also that includes ours. So if possible, I just goes to the first section. For this AD, because everybody knows this the conventional that AD processes from that complex that polymers finally to that methane. So anyway, today I just want to emphasize maybe this AD from that different viewpoint. The first one, for conventional AD, maybe usually we just focus on that solids, that waste treatment, but also for this anaerobic that treatment processes that can be used for wastewater treatment. And nowadays, for example, we can also use this that technology to treat that waste gases. For example, we can convert that CO2 that to methane. So anyway, from this aspect, just try to remember for these feedstocks that includes that solid, liquid, and also that gas. And also at the same time, just try to understand after this AD, and also 3 phases, that products, from this AD that including, that solid, maybe digested. This can be reused as the resources for fertilizer or maybe soil conditioner. And also maybe nowadays, for example, we can make that biochar from this digestate, and this biochar can be reapplied to this AD systems. For the liquid, for the treated water, this can be used for irrigation, directly, and also maybe this can be further treated for other reuse, for other purpose. And also for the gas phase, we can use this, methane to generate electricity, or maybe use, as the H. And also this can be upgraded to that biomethane. So anyway, here just to try to understand for the feedstock and also for this AD. Later I will introduce, that electrons that also includes that 3 phases, and also for the products, that 3 aspects. For each aspect, that includes that 3 phases. So anyway, for this AD, as we know, AD is that biological processes that should be one that ecosystem. So within this ecosystem, that includes, that organic carbon conversion. That this should be something like, material flow, and also because this ecosystem that includes diverse types of that microorganisms, they should, that communication, to each other. There should be some information that flow. Nowadays is especially that quorum sensing. That's also one topic. Here I just want to introduce for this system, because for some steps is that maybe requires that energy investment. So that requires energy input rather than produce that energy. That's why the syntrophic, or maybe collaboration among all these microorganisms is very important. So to provide this energy, that requires, that reduction or oxidation, for this organic carbon. So that's why this electron transfer is very important for this AD system. So the next, I will just focus on this electron transfer for this process. So anyway, for conventional one, as we know, usually we just focus on that, hydrogen. This should be, gas, and also that sometimes maybe formage. This also the electron carrier. This usually in the liquid, in the water phase, that soluble phase. And also nowadays we have introduced this direct interspecies electron transfer. So this one usually is just through that solid, that components. For example, that microorganism, they have, different that microbial components that electrons can be transported, from this one bacteria and to that, methanogens. So anyway, here just try to remember we have 3 that types or 3 phases of this, electron carrier. So this that one classic, that paper for that reported that DIT. So this DIT usually we just focus on that geobacter, and also this electrons can be transferred from that geobacter to that methanogens. So anyway, for this study here, I just try to mention several aspects. Even this paper, confirmed this DIT, but just remember here at the initial stage, even that, ethanol that maybe can be degraded, but the reaction rate is quite low, quite slow. At this stage, that's not so much that methane production, and only at the later stage, you can see here that methane can be degraded rapidly, and at the same time, that methane, produced. And also maybe certain amounts of that acetate can be produced. So from this study, another aspect is very important, because this figure used that fish that detected especially for these true that microorganisms, they need to form that something like aggregate similar to that granular or biofilm. Only under this condition, that electrons can be transferred. So this may be the reason, for this delay or lag phase, this methane production. And also for another aspect is very important, to understand that mass balance. Because from this study, they showed for this ethanol, especially 2 moles that can produce that 3 moles of that methane. So the mass balance through this calculation, they confirmed maybe DIET rather than that hydrogen or formate that electron carrier occurred within this reactor. Because also for these methanolians, they cannot use that hydrogen gas. So that's the reason for this study. They confirmed that DIT can occur between this gel vector and also this methanolians. So this paper is really very useful to understand that DIT. And following that, DIT has been, the focus, that is a very hot research topic in the field. So usually we just assume that DIT, this one, because it's just connect two microorganisms. There's no other chemicals or maybe other components to be involved. So this one usually that we consider this one is very high efficiency. And also this requires that less energy, and also that favored, and maybe long distance or short distance is depending on the microbial, that components. For example, some cable bacteria, they can transfer that electrons for several centimeters, maybe. So anyway, this something new about this, DIT. So usually, as I just mentioned, for this DIET, that usually we just use that microorganism, maybe that microbial that components, maybe salt chrome or maybe others to transfer this, that electrons. And also sometimes we can use that conductive materials that to replace these microbial components. This can also that transfer that electrons, and finally that can improve that system performance. So anyway, here just remember, at least we have 2 types. One is that microbial components that can transfer that electrons. Another one is that we can use that conductive materials. So another aspect for me, because initially we worked on this topic, but later stage sometimes we just question whether this DIT can really occur within that AD, because it's very important for microbial communities. If they want to conduct this DIT, they have to contact to each other. So this something like physical contact, that frequency, that should be quite important for this mechanism to occur. So anyway, from all the previous studies, for example, here, if we dose this conductive materials that including that GSA, granular activated carbon, or maybe magnetite, all of this. Usually, if we use these conductive materials, that one is that that can enhance the reaction rate. Usually, just remember this that one aspect. Another one just Another one is just try to remember here. If we have these conductive materials, the lag phase that can be shortened. So anyway, this the function of these conductive materials, and also that this may be through that DIT. Of course, for some studies, sometimes even without that maybe complex organic carbon, for example, that ethanol or maybe others, even only with that acetate, and that condition, sometimes that also increase the activity. So maybe the function of this conductive materials we need to consider one is that, syntrophic that reactions. Another one is just, maybe they have some other functions to increase the activity of that microorganisms. I'm not sure that should be okay or not. Yeah. So if it's okay, I just, continue. I think just recently, recent years, I read several papers, especially several papers from that professor. He wrote several papers about how to conduct that research in the microbiology that disciplines. I think it's very important for our environmental biotechnology or maybe environmental engineering. That is very important also for us to introduce some principles. For example, in our environmental engineering, usually we use that two types of that reactor configurations. One is that completely mixed, one is that plug flow, or maybe that SBR. So within these two types of that reactor configurations, there are different that substrate gradient. For example, for that plug flow, usually this substrate can decrease, from the influence to the effluent, from the highest, that concentration to the lowest. The lowest one that should be equal to that effluent. So and this, that high substrate gradient conditions that may enrich, this R strategist. But for this completely mixed that reactor, usually the substrate concentration is equal to that effluent. So on this condition, we have usually always that low that substrate concentration. And this condition that may enrich this K strategist. So anyway, now what's the R and what's the K? Usually for this R strategist, so they can grow very quickly, or maybe they can use that substrate fast, but they have low affinity for this substrate. But for K strategists, maybe they grow very slowly, their reaction rate is slow, but they can use that substrate more efficiently. So their affinity for substrate that should be, the value should be lower. So anyway, from here, we think maybe we can connect our environmental that reactor configurations to this, that ecological theory. Finally, we can just use this, theory to guide, our design of our, reactors. For example, for this AD, as we know, for the final that stage for methane production, usually we can, produce this methane through that hydrogenotrophic methanogens, or maybe we can through this, acetotrophic that methanogenesis. So if we just compare these two that pathways, usually for this hydrogen utilization, this reaction rate is quite high. So that may be for these methanogens, they are strategist. But for that, acetotrophic methanogens, they may be that K strategist. So if further, we just only consider that, acetotrophic methanogens, because usually, as we know, we have that two types of that, methanogens, for acetate utilization. And still, if we just use this R and K, we can check, for this methanol theta, this may belongs to that, K, but for this methanol succina, and this maybe belongs to that R. So this is the one example that shows maybe in AD that system we can introduce this RK selection theory. So anyway, for this AD, as we know, we want to remove that pollutants, and also we want to recover that energy. So if we want to introduce this DIET, that the first step we need to enrich this electroactive bacteria or maybe methanogens, and then the next step we can try to understand how to enhance that DIT. So here we think because Geobacter is one typical that EAB, that electroactive bacteria. So we think maybe we can just introduce this RK selection theory to enrich this gel vector, and further maybe we can improve that DIT process. So anyway, this is the background for this AD, especially I just introduced about this DIT, that new mechanism for electron transfer. So the next section I will introduce how to use that SRT and also that operational mode. This corresponding to that two types of that reactor configurations. I think for this one we just want to enrich that R and K strategist, especially for that gel vector. So for this purpose, we used SBR, anaerobic, that SBR, or this completely mixed. So we just use that different feeding durations. For this SBR, we only feed the reactor during that initial 10 minutes. But for this completely mixed that reactor, we just feeding the reactor for long duration until that settling that phase. And also for this study, we controlled that two SRT. One is that 25 days, one is that 10 days. So in total, we have that 4 reactors. So from this study, we used that ethanol. So we just increased the influence that COD concentration and finally to around that 4,500, and all these conditions only for this CFR, that something like completely mixed that reactor, and 10 days that cannot remove all the COD. This not the ethanol, maybe acetate and others. Anyway, for this one, because the 10 days SRT is not adequate for methanogens. Usually, for methanogenesis, maybe that SRT is more than 10 days should be better. But for other conditions, seems that can remove that COD efficiently. Even for this SBR, 10 days. So there's some difference for the system performance and different that SRT and also different that reactor configurations. So the next we just try to understand what types of that microorganisms within this different bioreactors. It's very interesting to find out, especially if we use this CFR that completely mixed or constant flow reactors, and both that SRT. So you can see here is the gel vector. This gel vector can be enriched efficiently within this that CFR reactors, but gel vector is not so high in this SBR that reactors. So this is something very interesting for us, because as we assumed, maybe for this CFR completely mixed, because within this reactor usually the substrate concentration is quite low, that may benefit the enrichment of this gel vector. Of course, just remember, as I just mentioned, for this CFR 10 days, there are still some effluent COD, but effluent COD is not the substrate of that ethanol. Because for this gel vector, they prefer to use that ethanol as the substrate. So anyway, from here that can confirm for this RK selection theory, seems it can be compliant to enrich this gel vector that are electroactive bacteria. So even for this methanogens, there are different types, but still for this SBR, seems this R strategist can be more enriched than K. So anyway, the story for this one is different, that maybe because the availability of the substrate, for example, for acetate is different from that ethanol, because the ethanol is just a feeding substrate, but the acetate is just produced from that ethanol. So that maybe the situation is different for methanogens from this EAB. But anyway, here that can confirm that CFR can enrich that gel vector. To further confirm the activity of COD removal or maybe methane production. This activity also shows under different conditions, especially for this CFR 25 days seems has the highest that reaction rate. So that may be connected to that gel vector enrichment. And also it's very important, as I just mentioned, it's very important to confirm the metabolic pathways for this ethanol degradation. So from this result, we can confirm for this CFR, you can see here, for these two reactors, just remember for this 25 days, seems the ethanol degradation only produce this acetate. There's no propanic acid production for this 25 days. But for this CFR 10 days, the propanic acid can be produced. So that means for this CFR, this SRT can affect this metabolic pathways. And also if we just compare, one is that CFR, one is that SBR. For both that SBR reactors, that propanic acid can be produced. So that shows this reactor configurations, or maybe that substrate that loading rate, something like that, that can affect that metabolic pathways for ethanol. So anyway, just try to remember here, as I just mentioned, for the effluent, even COD, concentration is quite high for this CFR reactor, 10 days. But the ethanol, that concentration is quite low. So that may indicate the why that gel vector can be enriched even under these conditions. So anyway, from this study, we can confirm maybe this gel vector can be classified as that K strategist. So this is very important. So for in future, if we want to maybe enrich that electroactive bacteria, maybe we can adopt this strategist, the strategy, or maybe approaches to enrich that K strategist to ensure always have that low substrate concentration. And also another one is just maybe SRT. This also very important, that because SRT can affect, in our discipline we use that FM ratio, because this food to microbial that ratio that may also very important. Later I will talk this again. But anyway, for this section, seems it's very useful to introduce that ecological principles to our discipline. So the next one, because I will introduce for this metabolic pathways, why? So because from the previous slide, I showed that propanic acid can be produced under some conditions. So anyway, just remember for this ethanol anaerobic degradation, that includes the conventional, that ethanol can be converted to this acetate. But for this metabolic pathway, that hydrogen gas can be produced. That's why we have to maintain low pressure of this hydrogen gas. So this is the conventional to acetate. And also, as I mentioned, for the principle, for this DIET process, usually this ethanol can be converted to that acetate, but at the same time, that they can just release that electrons rather than this hydrogen gas. So this the DIET process. Of course, these electrons can be used to produce that hydrogen, or maybe can be used directly by methanolens. The third metabolic pathway is just for this ethanol, they can react with that CO2 to produce that propanic acid. At the same time, of course, some acetate will be produced. So these the three that metabolic pathways for that ethanol that degradation. So for this study, still we use that SBR or maybe that CFR, this just two types of the reactor configurations to examine, especially for this SBR, as we just mentioned. This can just promote that propionic acid metabolic pathways. But for CFR, mainly that maybe that acetate or maybe that DIET. So for each that type of that reactor configurations, we also use that PSA. We hope this can enhance that DIET, but still is quite difficult to confirm. But anyway, here is the condition. We use that ethanol, still use that ethanol as the substrate. And also because we dosed this PSA, it's quite difficult, to control their concentration within the reactor. That's why for this study, we did not discharge that sludge from the reactors. So from the long-term operation, seems, for all conditions that COD can be removed efficiently because we did not discharge that sludge, so that some increase, maybe, of course, some still maybe just washed out from that effluent, but still SRD is quite high. And also we have very stable, maybe that, pH and others. So anyway, for the effluent, you can see here, under some conditions, especially for this SBR, that reactors, that effluent contains that propionic acid. So that means for this SBR reactor, still ethanol can be converted to that propionic acid. So this metabolic pathway should be enriched. But for this CFR reactors, after maybe around 5 days, and there's no propionic acid production in the effluent. So mainly is that there's no COD for the effluent for that CFR reactors. And also if we just check the metabolic pathways, and also for this SBR, both reactors, for this ethanol degradation, and also not only that acetate, but also that propionic acid can be produced. So this can confirm the phenomena from this long-term effluent that result. But for this CFR, both reactors, there's no much that propionic acid production. That indicates the similar that conclusions from the section 2. That means that reactor configurations can change the metabolic pathways. And also it's very important, if we check, this, because propionic acid can be produced in that SBR reactors. So that they also enrich that propionic acid oxidizing bacteria. So the activity here is much higher than this CFR reactors. And also CFR reactor does no much that methane production. This activity experiment also confirms that propionic acid degradation. Just remember, as I just mentioned, for propionic acid production, it requires that inorganic carbon, that is CO2, as the reactant to produce that propionic acid from this reaction. So that's why we also removed that inorganic carbon. And this condition seems, there are less that propionic acid that production and these conditions that also may be used as one evidence to confirm, the activity of that propionic acid metabolic pathway requires that, CO2. I think for this methanogenesis activities, usually for this hydrogenotrophic, this one, usually there's no lag phase. So I'm not sure, it's quite interesting. Usually, even for our reactors, if we take out, the sludge to conduct this activity experiment, usually for this acetotrophic methanogenesis, that requires some time. The lag phase usually is quite high. So this is something is different for batch experiments and also that reactor performance maybe. But anyway, under different conditions, maybe the activity is different. Usually for this SBR, these reactors, they have higher microbial activity compared to this CFR reactors. That because SBR reactors, they can enrich, hopefully maybe something like that R strategist. And also we conducted some experiments, to examine the effect of this PAC, this conductive materials. So usually if we have this one, they can enhance the activity, or maybe for example, that electron transfer chain that activity, or maybe conductivity. So anyway, that's no significant that improvement, but still the conductive materials can have certain effect for this microbial that activity. And also we used 16 or metagenomic sequencing to analyze the microbial community. And also for this result is similar to that section 2 for this CFR that reactor that can enrich of quite a high relative abundance of this gel bacteria. So this further confirms that gel bacteria is better enriched in these CFR reactors. This may still confirm gel bacteria could be that K strategist. But also it's very interesting, for this acetotrophic methanogens, we only enrich that methanol-3, the formula that methanol-theta, this K strategist. So this may be quite interesting, but still we don't know why. That maybe because I mentioned, because acetate is the substrate for these methanogens, but acetate maybe is always low. That may be the reason. For here, I will not introduce the details about this, metabolic pathways, but anyway, from this study, we can confirm for this SBR that reactors that can enrich this one type of that SRB, they can convert this ethanol to that propanic, acid. But for this CFR reactors, it's only have that, microorganism, they only convert that ethanol to acetate. So this is the difference. For this SBR reactors, because that propanic, acid can be produced, also they have, other metabolic pathways. So that should be some difference, for this too. So anyway, from this study, we can confirm that gel bacteria can be enriched, and also they can affect metabolic pathway, but still we cannot confirm that DIET. But anyway, this result shows some primary that conclusions, but it's not definitely to confirm that DIT. But anyway, it shows for this CFR that can enrich, especially for this gel bacteria, that may indicate that DIT. Further study will be required to confirm. So anyway, for these two sections, I just showed how to introduce that ecological principles to that AD systems and to enrich that specific types of that microorganisms, and then to enrich that different metabolic pathways. So finally, for section 4, I will try to propose some concepts. So for this concept that related to that Nexus approach. So anyway, first, for our environmental engineering or maybe biotechnology, if we want to achieve the purpose for engineering, we need to combine the principle and then to develop, that new technology. These two together, help to achieve the sustainability of our society or maybe others. But anyway, for our wastewater treatment or AD that systems that includes maybe the large scale, macro scale, and also finally to that micro scale. So anyway, basically, for the fundamental principles, that should be something like ecological or maybe microbial that principles. So it's very important to learn these microbial principles, including the fundamental of that microbiology or ecology. That should be very important. And finally, we need to know how to proper that selection of that functional microorganisms, and then further to regulate their metabolic pathways. So that's why if we go to that wastewater treatment, conventionally we just focus on that pollutant removal, and recently, especially during that 100 years that anniversary, during that time, lots of people just focusing on that resource that recovery from that wastewater, that including that water and energy and also maybe nutrients. But after that, I think the Nexus, that concept, has been introduced for water or wastewater treatment. Because for water, we will consider that water, not only water, we have to connect this water to ecology or maybe energy and also food, that health or maybe others. So it's very important, to just introduce this Nexus concept. Also for this pollutants removal, and especially for 2040, we will consider about an emerging compounds. This should be one challenge for the field. So for us, because around that 2020 at that time, we just think for conventional wastewater treatment, so it's based on that principle, but still we want to introduce that infrastructure based. We just separate different that reactors for different that reactions. This is the development for conventional one. At the same time, we think, can we just introduce something like microbial niche nexus? So, for example, for this different that infrastructure combinations, it's just introduce different functional microorganism. But for future, if we want to remove that emerging compounds, because most of the emerging compounds we don't know. Also, we don't know how to test or measure that's emerging compounds. So maybe based on unknown and also known microorganisms. So if we just combine these two, just to make sure that we can enrich more types of that microorganisms within this bioreactor, that maybe can help to remove known and unknown that pollutants. So this is the concept for this microbial niche nexus. We hope that we can introduce more types of that microorganisms and also more types of that metabolic pathways to remove that known and unknown pollutants. So for this one, still, if we go back to that RK selection theory, for example, now this is the one we know, usually for R, the R strategist, usually it has high that reaction rate, low affinity, but for K strategist, we have that low reaction rate but a high affinity. So, for example, if we just enrich both within one reactor, this can remove pollutants very rapidly. But for K strategies, they can ensure that low pollutants concentration in the effluent. So that's why if we can enrich this tool, that should be very good. Of course, if we want to extend this theory, for example, for this tool, if we this one is not expected, because this one has low reaction rate, low affinity, this should be not good for us. Of course, if we can, for example, in future we can use that synthetic microbiology, maybe we can design some microorganism. They can have very high that reaction rate, but they also has very high substrate affinity. So this one that may be something very interesting for us. So for this one, if we apply this RK to that anaerobic that system, for anaerobic system, usually I'm not sure it's different from that aerobic that activities large that systems. usually for anaerobic, we have very high that substrate utilization rate, or maybe that substrate fluxes. So on this condition, maybe I'm not sure, maybe the affinity is not that important under some conditions. So on this condition, maybe we can only focus on that reaction rate, use that flux-based concept to develop new technologies. So anyway, this some primary that concept for this RK and also that extension, how to further maybe apply this RK to maybe to design new that microorganisms. For this one, the reason for this one, because nowadays we can use that macro aeration for AD, because this can enhance the system performance, especially if we just introduce that low concentration of this oxygen that enhance the production of certain that enzymes. So anyway, that's why we can use this threshold. For example, we just maintain the oxygen concentration at 0, but at the same time, we can supply that oxygen. So finally, we just make sure the utilization rate and the supplying rate that equals to each other. And this condition, maybe we can just get some very good technology. But anyway, this RK strategies is just relative rather than absolute concept. So it's just for different microorganisms, they just compare to each other. So it's just relative to understand. This is why for this concept. So anyway, for AD, for anaerobic is different from that aerobic. So usually that the substrate concentration or maybe the reaction rate is quite high. So this is something like it's very interesting. As we know, for AD, the microbial growth is quite low, the activity is low. But finally, we can achieve very high loading rate for pollutant removal. But for aerobic, because the microbial activity is high, but the loading rate is quite lower than AD. That's because for AD we can enrich high biomass concentration, so that the strategy for microorganisms that should be quite different at these two conditions. So that's why also, for example, different that principles can be applied. One is that overflow that metabolisms. For example, for propionic acid production, as I just mentioned, this should be one type of that overflow than metabolism. So anyway, this something like these principles, from other disciplines, that can be applied. So another is, if we want to use that microbial niche, as I just mentioned, if we can enrich both R and K within one reactor, that may achieve this purpose. So if we want to achieve this R and K for our discipline, usually we can just focus on this food to microorganism ratio. So by control this parameter, maybe we can enrich both R and K within one reactor. So this still the basic knowledge in our environmental engineering, but how to understand this, how to refine our concept for this parameter, that should be quite important. So anyway, finally, for example, if we want to enrich R and K, still we can use that conventional that biofilm system. So within this biofilm, because we have different substrate gradients that can enrich different types of the microorganism, and also we can use that plug flow, just making sure there are some something like the conventional idle phase. So for this phase, there's no nutrients or no substrate for conventional functional microorganisms. But still for this one, this phase that may benefit the removal of the emerging compounds. So this, if we just use that R and K, that may refine our design of this, our conventional wastewater treatment processes, that including that AD. And also, if we want to achieve this purpose, it's very important to introduce some new principles. For example, one is that DIET, and also this functional that cable bacteria. This one is one example of that DIET. Because, for example, this different that reactions that can be connected by this long chain or maybe filamentous this cable bacteria. So that means the electrons can be transferred from long distance from this anaerobic to this aerobic that phases. So this should be something like this that should be quite useful. And also, if we want to understand the new principles, we need to understand the metabolic pathways. So it's very, very interesting, for example, for our disciplines, for sulfur or maybe nitrogen, because they have different that oxidation states. For this sulfur, that includes from that -2 to +6. So that different that intermediates or maybe different that reactions can occur within this system. So it's very important for our research or maybe others, not only to test the influent effluent, it's also very important to understand what's between the influent and effluent, what can be produced and what can be consumed again. So something like this one that should be quite important. So also for this concept, we need to introduce some new technology. So for me, because in the past I always consider about wines that animals, because for animals, they can grow very slowly, but finally, if they can be enriched, that high loading rate can be achieved, and this one is similar to that AD system. So because they are both that slow growing microorganism, but they can achieve very high that loading rate. Of course, for these animals, usually it's quite difficult to enrich. That maybe because they are autotrophic, but for methanogenesis, that should be heterotrophic. So that's why for AD, if we wait until maybe 20 days, that activity should be enriched. But for animals, that requires maybe longer time, to enrich this bacteria. So anyway, something like this one should be quite important to understand, how to develop new technologies, based on these principles. And also for our research, not only that experiments, but also this modeling, this both that should be quite important. Should be maybe just for we should connect this experiment to modeling and maybe that promote the further understanding of our maybe research result or maybe others. But anyway, this true that cycles from the concept to design the experiment and then get the results and analysis, finally propose some of the principles maybe in the field that should be quite important. Usually, I think it's very important, to combine these two. One is that experiment, one is that modeling, and also that lots of that papers mention about that methodology for research. This paper is just one example. For example, this is the objective, and for this research we need to propose some that hypothesis, and then we design the experiment. Hope we can achieve these outcomes. For this experiment, we should include different that maybe scales and also different steps. There should be sometimes that sequential. Sometimes we have this result have this feedback to the redesign. So if possible, for some students, you can just to read these papers. So finally, I just try to briefly introduce about this Nexus approach, because as I mentioned, nowadays for Nexus, this concept is very important. The first one, just try to understand this Nexus, that maybe including that connections or maybe linkages or maybe others. Sometimes it's just dynamic and fluxes sometimes you just, that steady state. So anyway, just try to understand this nexus. And also another one is very important for the name, for the nexus. For example, water, energy. Usually this just resources, resource that sector-based, or sometimes we just use that issue-based. So anyway, just try to understand there are two types of that nexus, concept. One is based on that water, food, energy. Another one is just maybe for CO2 that emission, or maybe carbon footprint, or maybe others. So these two types of that concept. And also for this nexus approach, so it's very important, finally, if we introduce this approach, we want to improve the system efficiency, not only just one aspect. We want to improve that all aspect. So maybe finally it's not the optimal for each one, but it should be optimal for the whole system. So this should be quite important. Of course, during this process, there should be some interactions. This should be considered. Finally we can provide different that outcomes. So this shows that at least 4 stages, for this nexus. Initially, it's quite simple, maybe just food to energy and water to energy. But nowadays we can just combine just more aspects. So this should be quite important, for you to understand, and what condition, what can be included, what should be included for this one. And also for this nexus, we need to understand, especially for different aspect. Sometimes maybe the synergy. We need to understand the trade-off between different aspect. For example, sometimes we just focus on energy. Sometimes we focus on that water. There should be some balance between these two aspects. Anyway, finally, we can use this method to understand the nexus, from that different aspect. For example, nowadays we just focus on sustainability, or maybe finally we just focus on that human environment, this nexus, because finally maybe we just need to protect our environment, and also that ensure the development of ourselves. And also that from the different skills, we need to consider the local, national, finally maybe global for this nexus. So anyway, just try to understand this one from the different skills. And also for this nexus, this concept, and we can connect this to different sustainable development goals. So anyway, just try to understand. Finally, we need to support what's the objective. That's also very important. So for this nexus approach, usually it's just something like concepts. For this concept, if we want to use this one to support our policy making, or maybe others, so we need indicators or maybe parameters, and finally we need something like value or score to assess the maybe the nexus, maybe the benefit, or maybe others. So this study shows one very good that example for this water, energy, and also food. So for each aspect that just support different that sustainable development goals. So anyway, for each aspect, they just propose at least 3 that parameters. One is that availability, one is that accessibility, and also sustainability. So they just produce that different that index. So finally, they can get this different. For example, this is the water, 3 dimensions, and also for this energy, and also for food. So finally, if we can use this something like that scores and also marks for each that index, then it's easier to compare. If we want to focus on which one, and then we can make proper decisions. And also from here we can just project future that development. So this something like how to use that Nexus approach. That's all for today's lecture. And finally, I just introduce this one because we have this survey. We want to understand what's the requirements in the field of this anaerobic digestion. So from the viewpoint of that, maybe professionals, maybe academia, or maybe students, for training or for research. So if possible, please help to provide your ideas about this survey. So that's all for my lecture. Following, if you have any questions or maybe comments, please, maybe we have 10 minutes, and after that, maybe Samira will introduce some basic knowledge about that metabolic based modeling for AD.
Wanna talk about metabolic modeling and constraint-based analysis in anaerobic digestion. For sure, all of you are familiar with anaerobic digestion ecosystem. There are microbiome in every ecosystem that you are familiar with. All of them can digest like the material in 4 steps: hydrolysis, acetogenesis, and methanogenesis. The result of this digestion is converting polymers and proteins to methane and CO2. The purpose of this presentation is to see if we are able to model this kind of ecosystem using metabolic modeling or not. First, I want to say what is a metabolic model. When we sequence genome of bacteria, then we can extract metabolic enzymes which are responsible for bacterial activity. Then we can connect it to a metabolic reaction. Using gap filling, we have a draft of a model which consists of some reactions and metabolites. After that, using literature review and knowledge gap filling, we can have a metabolic modeling. So the modeling consists of reactions which have reactant and products. We reconstruct it in a stoichiometric matrix which shows a biochemical network. These matrices are represented as the metabolic reaction that are happening inside an organism. And the coefficient of reactant and products in the metabolic network are the element of this matrix. Each negative number shows the reactant or consumption of reactant, and each positive number shows the production of metabolites. And we can represent them as a graph which age are reaction and node are metabolites. In this metabolic reaction, in this metabolic network, we should define something which we want to optimize. We call it objective function. Objective function is actually a function. It can be everything. In most of the case, we consider it to be biomass reaction, but it can be like methane production, it can be CO2, it can be growth rate, or everything which is related to our cell and we want to maximize or minimize it. This is the matrix, stoichiometry matrix that we talk about. And we have a flux vector. Flux vector is a vector that shows the rate of the reaction. The assumption that we consider in like flux balance analysis is steady state. It means that the concentration of metabolite doesn't change during the revolution of the network. So we should say S that V equals to 0. So we don't have any accumulation of mass in the network. It's kind of ensuring mass balance in the network. Then we add boundary condition to our system. This boundary condition limited the space of solution and give us a kind of polyhedral figure. Then we optimal this to reach one point, which shows the optimal value for objective function. For reconstruction parts, we have some software. For example, AutoCake, it use annotated genome to make an initial model. For then using manual curation in different databases, including Brenda, Cake, and MetaPsych, we have a curated model. Then we have kind of goes and back between FBA and curation of model to like validate model and have a model which is in high quality and meets the like experimental evidence. We have done it for Metantrix, which is one of the acid classic archaea. One other important point in metabolic network is reconstruction of biomass. As you can see, the biomass consists of carbohydrate, RNA, DNA, lipid, proteins, and different cofactors and metals and everything which is inside the cell. In most of the case, there is no like complete information, comprehensive information about the biomass, but we can use other model to reconstruct the biomass, use experimental data, or make some kind of guess about what is happening inside the cell, and then using experiments like confirm the guess. After that, we need to analyze the model, compare it with other model to see if like it is kind of relevant to other model. In this case, we have compared our draft with two published metabolic model, and we can see that most of the reactions in this model are like common reaction. It shows that the genome of almost all of the species are kind of similar. The important point is to be kind of noticed is that sometimes one genes or one reaction make a huge difference in the flux and in the function of the network. Then I want to explain a bit about flux balance analysis and its formulation and our assumption about steady state situation. If you consider metabolite A, there are some reactions that produce this metabolite and some reaction that consume it. For example, reaction B1 is producing it, and reaction V1 and V2 are consuming it. Each consumption is negative coefficient, and its production is like positive coefficient for the metabolite I. And it like determine the change in concentration of metabolite R. It is the same for metabolite B and C in our system. When we are talking about formulation, we can see that these coefficients are making stoichiometric matrix, and these like flux are the rate of reaction that we talk about it. So the concentration vector is equals to multiplication of a stoichiometric matrix and flux vector. When we are saying that we are in a steady state, it means that derivative term should be 0. So S that V should be 0. It is the assumption of flux balance analysis. In flux balance analysis, we can have different environmental condition. For example, in aerobic condition or anaerobic condition. It is a map of E. coli core model. And we want to see what will happen if we provide glucose and oxygen for E. coli. It is kind of aerobic condition which oxygen exists in the model. As we can see, using this color bar, we can see that the reaction which are active and the one which are not active. You can see that glycolysis, also TCA cycle, and some exchange reaction are active when the situation is aerobic. But when we remove oxygen from the medium and environment, we can see that TCA cycle is not active anymore. And instead, the fermentation part is working. It shows us the difference between different condition, and we can like use it in modeling anaerobic digestion. For dynamic FBA, we assume that the kind of linear relation between like change of concentration and the initial concentration, and we have kind of exponential growth for our model. Using dynamic FBA, it is like jumping from one steady state to another one to see time difference between different conditions. We want to show a respirofermentative organism when it's kind of using FBA to see what is happening to a bacteria. It is a bacteria that can operate both in like aerobic and anaerobic condition. So the bacteria use glucose and produce acetate. This is the concentration of an enzyme which is responsible for fermentation. When we plot the flux as a function of growth rate, we can see that in initial like step of working, the concentration of acetate is very low in the medium, and glucose uptake is kind of increasing. And we can also see that the enzyme expression is also high. But when the glucose uptake reach its maximum value, acetate secretion is also started, kind of using glucose and turning it to acetate. Then we want to like use kind of communication between these bacteria. One of them is able to produce acetate out of glucose, and the other one is not able to do that. So we close the reaction responsible for production of lactate and acetate, which are the product of fermentation. So now we want to have two models and do dynamic FBA for this community. As you can see, the biomass production for these two bacteria is different because one of them is kind of defective, not able to secrete lactate and acetate. The biomass production difference is shown here, and also the uptake of glucose and acetate for these two bacteria is shown here as a function of time. We can see that production of acetate for the bacteria increase at the time that glucose concentration is in a good level, and then decrease since the glucose is finished in the medium, so the bacteria is not able to produce acetate. And we can see that glucose and acetate production in the medium, we can see the concentration of glucose is reduced to reach to 0, and acetate production is also in this way. This acetate production is the result of the first bacteria which is able to produce acetate. And this kind of analysis is possible using a constraint-based modeling. But if we want to talk about challenges, we should say that there are always some annotation that are not in high quality. Sometimes we need the kinetic parameter, for example, for growth rate, which we consider to be linear. Sometimes it is not possible to make that kind of assumption. We also are able to integrate omics data to our model and process data according to different environmental conditions. But the omics data are not always available for different microbial community. And it is important to know to mention that sometimes some bioinformatic analysis using machine learning and data analysis help refinement of model, which need to be considered. The key points from this presentation: we are able to model metabolic modeling, it's kind of a bridge from microbial genes to system level behavior. We are able to use a different constraint-based approach like FBA and dynamic FBA to know more about the kinetic of the system and the flux of reaction. We are able to make kind of relation between different nexus like carbon, nitrogen, and energy cycle. And it is important to do some kind of model designing to have more optimized and efficient system in anaerobic digestion. Thank you for your attention. If you have any question.